freshman at Chapman University Schmidt College of Science and Technology, majoring in biological sciences. And the research that I'm involved with at Chapman's Pharmacy School is on interfering with mTOR signal transduction in breast cancer cells. Now, what does this mean and how do we accomplish this? We are treating the cells with specific doses of the drug everolimus. Everolimus is a rapalog drug, which means it's a member of the rapamycin family, which functions as an immunosuppressant. The drug was originally used for organ transplant. However, the symptoms that the patients quickly got after getting the drug donned the drug's nickname the Dracula drug because they experienced foaming around the mouth, pale complexion, psychological trauma, a destroyed immune system. It was terrible. So they took the drug off the market. However, we are using the drug in this experiment because it specifically targets the protein mTOR C1, which I will go into later. There are 3 genes found that correspond to rapamycin, and one of those genes is mTOR. mTOR actually stands for mechanistic target of rapamycin. The breast cancer cells in this experiment are especially difficult to treat because they're triple-negative cancer cells, which mean that they do not respond to hormones such as progesterone, estrogen, or HER2. This means that they do not respond to traditional drugs. And the 2 triple-negative cancer cells that we are working on in this experiment are MDA-MBA431 wild type, or wild type 431, and MDA-MBA231 wild type, or wild type 231. Wild type 468 is a primary cell extracted from a person with breast cancer, and wild type 231 are, if you look at them under a microscope, they're sharp-looking cells with a very high proliferation and maturation rate. If somebody with breast cancer is found with wild type 231 cells, they can reach up to stage 4 cancer within the span of a week. We choose to silence these cells instead of inhibiting because the cells will learn to use their other intrinsic capabilities to survive, and they become immune to small molecule drugs very fast. Inhibition will cause an increase of everolimus side effects. To understand how signal transduction works in oncogenic cells, here's an image of the regulation of the cell cycle through protein signaling of different receptors. So all of these complexes around here are protein complexes, which send signals back and forth with each other. And along the membrane of the cell at the top are protein receptors, which also functions as hormones. All of the proteins with an arrow going away from it, such as these, are called upstream proteins, and all of the proteins receiving an arrow are called downstream proteins. The JAK receptors on the cell's membrane are hormones, and G-beta gamma, which is depicted on the right here, are G protein coupled receptors. And in cancer cells, they become highly active. The cell cycle starts with PI3K here, and PI3K acts as a first messenger. It is very active in normal and cancer cells. PI3K goes to PIP3, and PIP3 gets a glucose molecule attached to it, and this acts as a second messenger. PIP3 is also very active in cancer cells. PIP3 then goes through several activations before it eventually reaches AKT in the center here. AKT, otherwise known as protein kinase B, it acts as the control center of the cell. It's responsible for activating protein complexes as well as acting as a proliferation messenger. The main focus of this project is on the complexes mTOR C1. mTOR decides how much the cell will proliferate. It acts like a sensor, and there are 2 complexes in mTOR. There's mTOR C1, and there's mTOR C2, also depicted on the bottom here. Only the growth factor can activate both mTOR C1 and C2. mTOR C2 focuses on cell survival and cytoskeletal organization, and mTOR C1 focuses on metabolism, growth, autophagy, which plays a role in cell degradation, and biosynthesis, which is the production of molecules within the cells. mTOR C1 has an important component that we are studying in this experiment called MLST8. MLST8 acts as a linker. There's not a lot known on MLST8, but we guess that it is highly sensitive to cell lysis pressure, which means that the cell will burst. And when little pieces of the cell blow away, they can blossom into cancer. So therefore, extracting MLST8 alone is a big challenge. It has 7 WD-40 repeats, which is a mixture of tryptophan and aspartic acid, and heat shock sequences, which act as a linker for the whole complex to come together. MLST8 gets activated by a phosphate molecule, and then once it's activated, it sends signals and gets attached by 3 other components which form bonds with each other. In previous studies, omission of MLST8 led to the omission of the entire mTOR C2 complex. The mTOR complexes are made up of different proteins such as FKR, MLST8, and STN. However, what distinguishes the two is that mTOR C1 has a Raptor protein, depicted here, and mTOR C2 has a Richter protein, depicted here. And the Richter protein is significant because it has strong binding properties and plays an important role in cancer cell progression. In gastrointestinal tract cancers and breast cancer, the PIP3K AKT mTOR pathway is overactive, which means that cell proliferation will occur. So you may be thinking, why don't we just block this pathway before the possibility of cancer is even an option? Well, this pathway is very important in stem cell differentiation, which means that if you block this pathway, cells such as muscle and tissue won't form. So that means that you would have to lead an extremely cautious life to avoid minor cuts and injuries. So essentially, we want to stop the overactive proliferation of the cell without affecting its homeostatic properties. Our hypothesis is, because MLST8 acts as a linker in protein signaling between the pathways AKT and mTOR, through silencing MLST8, we can inhibit cancer cell progression. So this project is unique because it's the first research project of its kind to be focusing on breast cancer cells specifically. And rather than focusing on complex inhibition, we are focusing on formation inhibition, which means that we are trying to stop the complex from getting formed initially. And we're also focusing on the subcomplexes MLST8 rather than the whole mTOR complex. So, cells are smart, and they have a high plasticity rate. So if we inhibit the pathway from AKT to mTOR, the cell's cytoskeletal structure will actually change shape, and the cell will find a different way to get from AKT to mTOR. So it's like taking the side streets instead of the freeway. And once the cell changes shape, there are many, many different pathways that it could possibly take. So because of this unreliability and inconsistency of how the cell functions, how is one to track the cell's next configuration tactic? So that's why our focus is on drug delivery. And one side street in particular that has a common presence in this pathway is called HER2. And HER2 is a receptor on the membrane of the cell depicted here. HER2 is a member of the EGFR family. And EGFR inhibitors are actually the most useful drug because it responds to the existence of a growth factor. EGFR is also triple positive, which means that it does respond to hormones such as progesterone and estrogen. EGFR inhibitors are also the most useful drug because it finds the growth factor, it adds an inhibitor, blocking nutrients from entering the cell. And HER2 will actually, to streamline nutrients into the cell, HER2 will grab a blood vessel, bring it into the tumor, so the nutrients can make the tumor flourish and thrive. So that's why some chemotherapy drugs are EGFR inhibitors, which means that it will block that nutrient streamline. The cell will, so once the inhibitor is attached, the cell will then die from starvation. As the tumor cell forms, HER2 produces signals that bring in the new blood passage. But interestingly, a cell may start with 0 HER2 receptors and grow to form many. Cells are smart because of their keen adaptations to these environmental factors. We are treating the cells with LD50, and this is the lowest dose of everolimus that can kill 50% of the cell's population. We look at the LD50 of different cell lines and determine what dose can kill this percentage of cells? One of the methods that we perform in this lab is resistance induction, both gradual and shock. So through gradual resistance induction, we actually train the cells to become resistant to the drug everolimus. Continuous exposure to everolimus causes the cells to learn, adapt, and become resistant to its effects. First, we treat the cells with the lowest concentration of LD50. We repeat the experiment 3 times over the course of 3 weeks until we reach 1.5 times LD50. And with this experiment, wild type 468 survived. Through the shock method, we test the cells for their intrinsic capability to survive. So instead of gradually teaching them how to become resistant to everolimus, we look to see which cells have this intrinsic capability. So first we acquire LD50. We treat the cells with LD50 1.5 times more, which kills 75% of cells until they become resistant to everolimus, which is 2 to 3 times higher than the primary LD50 reading. The highest dose exposed was 40 micrometers. And with this experiment, 2 cell lines survived, and that was 231 wild type and 231 mutant. And after these methods, we perform primer designing and a PCR panel, or polymerase chain reaction. 48 different targets were placed in this panel, all of which play a critical role in the AKT mTOR pathway. And we plan to track the cells' shift and shape in active pathways through the over and under expression of these targets. We conduct PCR to study the quality of gene expression after silencing the gene with siRNA. And siRNA is a designed RNA strand used to target a specific mRNA for silencing. And we specifically use real-time PCR to show us how many genes we have, indicating that an overexpression of a gene links to a certain protein. So through culturing, we make the cells resistant to the anti-cancer drug everolimus. Our design panel of different primers are what make the cells resistant. Our hypothesis is G-beta-L stops mTOR C1 formation and hides downstream proteins like P70 and 56K. Upstream proteins lead to the activation or inhibition of mTOR. And the primers that we use in this experiment are what are called housekeeping genes because of how often they are used in the lab. And these include beta-actin and HPRT1. So this is some data from the experiment. This is from the cell line MDA231 wild type using the shock method. So down here you can see that the trials we made with the no treatment, trial 1, trial 2, trial 3, trial 4 subsequently. And the graph shows the cell viability percentage to the concentration. So this shows the resistance in cell lines through our different treatments. And at each treatment interval, we add 1.5 times the amount of LD50, let it sit for 72 hours, passage them, or culture them, which means that we make the cells grow. And then we continue adding 1.5 times of the drug until the 4th treatment. And this is a graph from the cell line 231 gradual method. And so, as you can see over here, we start with 10% of the LD50. We treat them with 10% LD50 for 2 weeks and then increase the dosage to 20%. Then after passage of the cells, we divide them into 2 groups. One group has mRNA extracted, and the second group is then receiving the everolimus medication, either shock or gradual. Once we extract the cell line, we do PCR, and then from the divided group, we seed the cells in a 96-well plate at different ranges of medication, wait 72 hours, and then we add CCK color dye and acquire a new LD50. Each number on the graph shows the LD50 after each treatment. And we continue the experiment until we see notable resistance, or 3 to 4 times of the primary LD50. So after resistance has been induced, we perform PCR, and using the same PCR panel that we used on our silent cell lines. So as we can see here from the key, there is an overexpression of the HER2 gene. And HER2 as a triple positive complex, having an overexpression is very interesting. And we will perform a Western blot to further show the reproducibility of these results. So overall, the reason that we silence the cells with everolimus is we're trying to make the cells resistant to the anti-cancer drug. So once the cell becomes resistant to the anti-cancer drug, we can see how the cell shifts its shape and uses different pathways. And we want to find something in common between these different pathways that the cell decides to take. So from the PCR panel, we have found that a common pathway that it takes is through HER2. And we think that HER2 plays an incredibly important role in cancer cell proliferation and tumor formation. So in the future, we want to run a Western blot on resistance-induced cells to show the reproducibility of our experiment, as well as do simultaneous silencing of the MLST8 complex and other components of the same or neighboring pathways. And the next phase of the experiment is to analyze the structure of mTOR C1 and mTOR C2 in the absence of MLST8. I would like to thank Dr. Hamid Reza Montazeri Aliabadi for letting me into his lab last semester and this semester, as well as my research mentor, Ramina Nuri, and the Chapman laboratory personnel for assisting us in the experiment.